You are all welcome to my YouTube channel. And please, if you know that you didn't join and subscribe to my YouTube channel, please try to do that. And it is very, very important to join and subscribe to the YouTube channel, which will actually give you more chances to have access to our videos and multiple choice questions. So please, if you are coming for the first time and you know that you didn't join and subscribe to this YouTube channel, please try to do that. That it would actually increases the global recognition of the YouTube channel. Please subscribe and join. So in today's video, we are going to look at the transport, or in today's lecture, we are going to look at the transport of materials across cell membrane. And my name is Mukhtar Aliyu Ikara. And of course, we need to ask yourself this question. We know that the body is made up of cells, tissues, organs, and then system. Then we have the complete living organisms. So we eat foods, we digest foods, we take drugs. But the question is where these food substances, where the nutrient from the foods, where the drugs are utilized and metabolized in the body. Strictly speaking, the nutrients, the drugs that we are taking are utilized and metabolized in a cell. So after we take foods or we take drugs, the drugs and the food's nutrient will be digested, transported, and then absorbed. And after absorption, the drugs and the nutrient will be in the blood. But what next? The blood, of course, is a media for the transportation. So it transports nutrient and drugs to the target cells. So when you transfer them to the target cells, what happens? The nutrient and the drugs must get into the cell. So how the nutrient and the drugs get into the cell before they metabolize and utilize by those cells is what we are going to look at in this lecture. That how a nutrient, like let's say glucose, after we take carbohydrates, we digest carbohydrates, and then after the digestion, we transport it, we absorb it into the blood, and then from the blood, the glucose will be transported to the cells that are needed. So now after they transported to that cells, how the glucose molecules gets into the cell. And that is what we are going to look at in today's lecture, the transport of materials across the cell membrane. So introduction to cell membrane transports. So cell membrane is a crucial barrier that regulates the movement of substances in and out of the cell. So this cell membrane, it has a double layer and it is a barrier. And the main functions of this cell membrane is to regulate what will get in and out of the cell, because there are so many substances that are not needed in the cell. So cell membrane will regulate that substances, and there are substances that are surely needed in the cell, and then of course they will be allowed to get into the cell. So the cell membrane, it's playing this important role, is like a barrier that control what will get into the cell and what will get out of the cell. Assuming now it is a bacteria that want to get into the cell, so it is the main function, or it is the function of the cell membrane, decide whether that bacteria will get in or will not get into the cell. So the cell membrane is very, very important. It's like a wall that regulates what will get in and out of that particular cell. So generally, after we take drugs, like for example, we take drugs, and generally drugs are metabolized in the hepatocytes, that is the cells of the liver. So after the blood reach into blood circulation, then the drugs will be transported the hepatocyte. Then after it transported into the hepatocyte, then the membrane of the hepatocyte will now control the transport of the drugs from outside of the cell into the cell before it is fully metabolized. So of course, we know that the drugs is metabolized in the smooth endoplasmic reticulum of the cell. So drugs is utilized or is metabolized in the smooth endoplasmic reticulum of the cell. So it means that if you have the drugs outside of the cell, it must be transported from that outside of the cell into the cell for it to get into the smooth endoplasmic reticulum of the liver cells, which is a hepatocyte, before it is being processed, metabolized, and then excretes. So of course, this is a cell membrane. If you can see clearly, this is how the cell membrane looks like. This is how the cell membrane looks like. It is a double layer. This is the first layer, and then this is the second layer. So, like for example, this is a sodium ion, one of the materials. That is the metal ions. So this is, look at how it is trying to get into the cell. So the transport of substances across the cell, it is actually controlled by the membrane, and this membrane is highly rich in protein carriers and protein transporters that are fully embedded into the cell membrane. And some of these, like for example, this is a protein channel. This one is also a protein channel. So if you look at this protein channel, it is fully embedded into the membrane of the cell. So this is a typical example of integral protein. If you could remember from our previous lectures, we actually explained that integral proteins are the proteins that are fully embedded into the cell membrane, while peripheral proteins are the proteins that are partially or transiently attached to the membrane of the cell. So of course, in this case, we have a carrier and the channel proteins, which is also very important in controlling what will get in and out of the cell. So then the next point is the cell membrane. It consists of a phospholipids or phospholipid bilayer with embedded proteins. So this is a typical example of embedded proteins. And of course, this is a phospholipid bilayer. This is the phospholipid bilayer. This is the first layer, and this is the second layer. That is why it is called phospholipid bilayer. And of course, this head is hydrophilic, and this head also is hydrophilic, while all this part is hydrophobic, which is a non-polar region of the membrane. So the polar head of the membrane, or the polar head of the phospholipid, it is the hydrophilic layer where it attaches and interacts with water molecules, while the non-polar component, which is here, it is a hidden part. So it's hidden because it doesn't interact with water molecules. And of course, it has embedded proteins, and this is the embedded protein, which is an integral protein, and at the same time is considered as a channel proteins where biomolecules get into the cells cross it. So then understanding how materials cross this cell membrane is essential for studying cellular function. So if you are able to understand how these materials get in and out of the cell, or how these materials get across the cell membrane, it is very, very important in studying cellular function. So what are the types of transport mechanisms? So we have two major types of transport mechanisms, although in some textbooks, they will tell you that there are three major types of transport mechanisms. Number one, we have passive transport, and number two, we have active transport. And some other textbooks, the third other types of the transport mechanisms is bulk transport. So we have passive transport, active transport, and bulk transport. But in this case, we are going to concentrate on passive and active transports. So passive transport is actually a movement of molecules that doesn't require energy. So a passive transport is a type of transport that allow the materials to get in and out of the cells without the need of energy. So it doesn't require energy to do that. Then we have active transport, which requires ATP to move substances against their concentration gradient. So in the case of active transport, it actually control the transport of substances, but with the use of energy. So it uses ATP, which is the energy for it to transport substances across the cell. So in the case of active transport, it requires energy, and passive transport, it doesn't require energy. So this is a basic explanation of these two concepts. Passive transport does not require energy, while active transport require energy. One, and second, in the case of passive transport, the molecules are actually moving from the region of higher concentration to the region of lower concentration. That is where the molecules are lower. Where the molecules are lower from the region of higher. And in the case of active transport, it actually transports substances against concentration gradient. That is, the molecules are moving from the region of lower concentration to the region of higher concentration. So these are the two main basic differences between these types of transport. And as we continue with the lecture, we'll also see how they further differentiate themselves. And then each mechanism plays a vital role in maintaining homeostasis within the cell. So one very interesting thing is that these two types of transports, they are very important, and they are all trying to maintain homeostasis. That if the amount of or if the concentration of glucose is supposed to be within a certain number, then it should be maintained within that numbers. And if it reach that numbers, then there will no be longer transportation of glucose, whether in and out of the cell. So that is why most of these physiological parameters, they have their normal range, and if they are above that or below that normal range, it means that there will be a problem. So one of the very important mechanisms that trying to make sure that all these physiological parameters are maintained within the normal range is also transport mechanisms. Like if the amount of creatinine is supposed to be 2 milligram per mil in the blood, then it should be maintained within that. And if the amount of the glucose in the red blood cells is supposed to be 1 milligram per mil, then it should be maintained within that. So therefore, once it reaches that level, and the red blood cells or the membrane of the red blood cells will no longer allow the transport of the glucose into the red blood cells. So transport mechanisms is very important in trying to maintain homeostasis, that is regulation of the internal environment of the cell. So as I said, actually active versus passive transport. So we can see clearly actually from this image, we can see that in the case of active transport is a movement against the concentration gradient, and it uses ATP. So if you look at it, look at the molecules here, then of course, this is a molecules / ions. So it can be a molecules or ions. And of course, this is also a molecules or ions. So you can see clearly these molecules / ions, a rate, their concentration outside the cells here is low, and then their concentration inside the cells is high. So that is why we said against concentration gradient, because the movement is happening from the region of lower concentration to the region of higher concentration. So this is how the molecules are fastened from the region of lower concentration to the region of higher concentration, and that is why it is called active transport. And of course, ATP is utilized, that is ATP here, so energy is needed. So energy is hydrolyzed. So this is outside the cell and this is outside the cell. So the molecules are moving from the region of lower to the higher concentration. That is why we said concentration against concentration gradient. And then at the same time, we have passive transport. So this passive transport is divided into two. We have simple diffusion, and then we have facilitated diffusion. So in the case of simple diffusion, the movement along the concentration gradient without energy. So you can see clearly, look at the diffusion particles here. So their concentration outside the cell is higher than inside the cell. And of course, it is moving from the region of the lower concentration, from the region of higher concentration to the region of lower concentration. So in the case of passive transport, the molecules are moving from the region where the concentration is higher to the region where the concentration of the molecules is lower. So that is in the case of simple diffusion. And in the case of facilitated diffusion, it is also the same applied as a simple diffusion, where a molecules or ion gets into the cells from the region of low or of higher concentration to the region of lower concentration. But in this case, in the case of facilitated diffusion, two things are very important. Either channel protein or protein carrier, they are needed for the transportation to take place. So it needs a channel, which is a protein channel for the molecules to get in, or a protein carrier like a cargo, like a car, to carry the molecules and get them into the cell. So in the case of facilitated diffusion, protein carrier as well as a protein channel are needed. They are needed for the transportation to take place. So this is actually in the case of the passive transports and active transports. So let's look at the passive transports more closely. So passive transport occur when substances move from an area of higher concentration to an area of lower concentration, as explained. So from higher to the lower, and then this passive transport is actually divided into three. We have diffusion, osmosis, and facilitated diffusion. So these are the three types of passive transports. And then cells rely on passive transport for the movement of small non-polar molecules and water. So generally, substances or molecules uses smaller molecules and non-polar molecules as well as water uses this passive transport mechanism to get into the cell. So ladies and gentlemen, if you are following my YouTube channel and join this channel, please, I wanted to put this answer in the comment section. Give examples of two molecules that get into the cell using passive transport, and also examples of non-polar molecules that get into the cell. But although in the case of non-polar molecules, let me just actually give this example so that at least, because it is very important. So examples of non-polar molecules that get into the cell using passive transport, like drugs. So generally drugs are lipophilic, and at the same time, the membrane of the cells is a lipid bilayer, so it's made up of lipids. So generally drugs are actually designed as a lipophilic, because when we are in disease, we need to get drugs to get into our cells as fast as possible. So that is why most of the drugs, they are designed as a lipophilic, so that they can easily dissolve in the cell membrane and then get into the cell. So it doesn't need any extra energy. It doesn't need any protein carrier. It just gets into the cell using a simple diffusion. So that is why most of the drugs are designed as lipophilic, so that they can easily get into the cell, or easily dissolve on the cell membrane and get into the cell. So this is actually rationale behind designing drugs as a non-polar, as a lipophilic molecules, because if they are polar, then they need an extra energy and extra effort before they get into the cell, and as a result of that, it can even lead to the crisis and even death of the patient. So that is why it is very, very important to design drugs as a lipophilic molecules. So actually, let's look at the facilitated diffusion, which is one of the passive transport. So in facilitated diffusion is the passive movement of molecules or ion across a plasma membrane by a means of a transport protein in the plasma membrane. So it can be a transport protein or a carrier protein. It can be a transport proteins or a carrier proteins, and at the same time, it can be a channel. So a protein channel and carrier proteins are needed to get the molecules into the cell. So of course, if you can look at from this image, you can see that of course the smaller molecules, the particles or the molecules here have their higher concentration here, while the concentration of the molecules here is lower. And of course, because their concentration is higher in the extracellular space and then lower in the intracellular space. It means that using passive transport, they can get into the cell. But because here we are talking about passive transport, then protein carrier and then carrier proteins, they are needed to get the molecules into the cell. So please, as I said, I wanted you to give me examples of small molecules. And apart from the drugs that I mentioned, also I need another examples of one non-polar molecules that get into the cell using passive transport. Please, I need that answer on the comment section. Thank you. After you watch the video, please answer this on the comment section. And please, I always encourage you to try to subscribe and join the YouTube community. And then the next thing actually is diffusion, which is one of the examples or one of the types of passive transport. So diffusion is the movement of molecules from a region or from an area higher concentration to an area of low concentration until equilibrium is reached. So in the case of diffusion, actually the molecules are transported from the region where their concentration is high to the region where their concentration is low. And it is a spontaneous process, because why we said it is spontaneous, it means that it doesn't require any external agent, it doesn't require any cellular energy before it moves the molecules. And then at the same time, small molecules, uncharged molecules such as oxygen and carbon dioxide typically use simple diffusion to cross the cell membrane. So you know that actually oxygen is one of the air that we are needed for cellular metabolism to take place, while carbon dioxide is the end product of cellular cellular respiration. So we need this diffusion to get the oxygen in and carbon dioxide out of the cell. So small molecules and uncharged molecules are transported using simple diffusion. So this is actually a typical example. This is a diagram showing how the diffusion mechanisms take place. We can see here diffusion is actually a movement of particles from higher to a low concentration. So if you can see clear here, we have a dye molecules here, and then we have water molecules. And of course, you can see that the dye molecules is highly concentrated in one region. Then of course, the movement to low concentration. So the molecules start actually spreading. It starts diffusing. So after that, then it diffused eventually, it's diffused evenly, and then equilibrium is reached. So you can see that the molecules are diffused evenly, and then it is actually spread, and of course equilibrium position is reached. So this is diffusion. It's also applied, typical examples of this diffusion is like perfume. When you spread a perfume, the perfume can be actually moving from the region of higher concentration to the region of lower concentration. That is from you that have the perfumes to the region where there is low concentration of that perfume. So it will start spreading, diffusing, and then until it's evenly distributed. So of course, this is a simple diffusion. And then the next thing is osmosis. So osmosis is the diffusion of water molecules through a selectively permeable membrane. So osmosis and diffusion are the same thing, until that in the case of osmosis, a selectively permeable membrane is needed before the molecules diffuse or transported. So water moves from area of low solid concentration to the areas of high solid concentration to balance solid levels. So if you have a solid particles, where the solid particles have a lower concentration, and then of course the water molecules will pass from the region of low solid concentration to the region of high solid concentration, so that the balance of the solids will be achieved. Then this process is vital for maintaining cell-to-growth ratio and overall cellular integrity. So it's very, very important in maintaining the integrity of a cell. because if like one part of the cell is highly rich in the concentration, like for example, if outside of the cell you have enough concentration of water and inside of the cells you have high concentration of solid particles, like let's say salts. So of course you need a water molecules that can get into that cell so that you make sure that the uniformity as well as the equilibrium position is achieved in order to maintain the levels of the solid particles. So if that is not achieved, so there is a high chances the overall integrity of the cells will not be maintained, and which can actually at the end lead to the damage and then death of the cell. So this is a typical examples of how the molecules actually move from the region of lower concentration to the region of higher concentration. So look at it here, the concentration of water here is very high. So therefore the molecules here have a lower concentration. And then of course here the molecules have a higher concentration. So therefore the water molecules will now move through this semi-permeable membrane to the other region so that to achieve equilibrium. So the same concentration is achieved by actually neutralizing the concentration of solids levels using the water molecules. So that is actually osmosis. So the next thing is facilitated diffusion. So facilitated diffusion involves the use of specific transport proteins to help move substances across the cell membrane. So it needs a transport proteins and carrier proteins to transport substances across the cell membrane. So this mechanism is crucial for larger or polar molecules. So see, facilitated diffusion, you see, so you try as much as possible within the facet transport to try to differentiate between simple diffusion, osmosis, and facilitated diffusion. So in simple diffusion, small molecules and non-polar molecules are transported. And then in the case of facilitated diffusion, larger molecules or polar molecules that cannot be easily passed through the lipid bilayer, then it's now actually moved or it gets into the cell using the facilitated diffusion. So the common examples include glucose transport and ion channels that regulate ion flow. So glucose transports as well as the ions molecules can actually, or ions like sodium-potassium can be moved or passed through the cell membrane. So this is actually a typical examples of facilitated diffusion, and look at it here. So this is a typical examples of a channel protein. This is a channel proteins where the molecules can get into the cell. You see the concentration of the molecules here is higher, while the concentration of the molecules here is lower. Or like for example, in the case of these molecules, it can only get into the cell using a protein carrier. So ladies and gentlemen, please don't forget to try to subscribe and join the YouTube channel. Okay, Khadija, thank you for joining the channel. So thank you. Okay, Peter, you are also welcome for joining the YouTube community. So this is actually for the passive transport. So the larger molecules or polar molecules can actually be transported using this facilitated diffusion with the aid of protein channel and protein carrier or carrier proteins. And of course, the molecules are moving from the region of higher concentration to the region of lower concentration. And typical examples of molecules that can utilize this facilitated diffusion is glucose molecules and ions like potassium and sodium. So the next types of, although we have cotransport, so a cotransport is also a part of simple facilitated diffusion. So a cotransport is a process that moves two substances across a cell membrane at the same time using a protein or protein complex. So a cotransport is a type of transport that involves the transport of two molecules across a cell membrane at the same time. It can be in opposite direction or it can be in the same direction. So there are 3 types of cotransports. So in some textbooks, they will tell you that there are only 2 types of cotransport. But here we have 3 types of cotransport. So in the case of symport, which is one of the cotransport, is actually when both molecules move in the same direction. So when you have 2 molecules are moving in the same direction across the cell membrane, so that types of transport is called symport transport. So examples of molecules that move across the cell membrane using symport is glucose and sodium ions. So glucose and sodium ions get into the cell membrane at the same time in the same direction. Then the next one is antiport. So in the antiport is when both substances move in opposite direction. So when you have 2 substances moving in opposite direction at the same time, these types of transport is called antiport, like sodium and potassium ions. So if potassium or if sodium is getting into the cell, then the potassium will be getting out of the cell. Like generally, 2 sodium usually get into the cell, while 3 molecules of potassium ions get out of the cell. So they are moving in opposite directions. So as sodium getting in, then the potassium will be going out. Then we have uniport. So a uniport is when only 1 molecule pass across the cell membrane. So let's look at this example. So if you look at the case, in the case of this cotransport, look at the symport. So in the case of symport, look at this red ball and this ball. Look at them. They are getting into the cell in the same direction. You see, they are getting into the cell in the same direction. So look at them and at the same time. And then of course, for the antiport, you see, as this ball is getting into the cell, and then the red ball is getting out of the cell. So that is antiport. Then in the case of uniport, then is when you have only 1 molecule is getting in from outside, inside of the cell. So if you look at this cotransport, is actually involve the use of a channel protein and carrier protein. So that is why we said cotransport is still under facilitated diffusion. So the next thing after the cotransport is active transport. So what is active transport? So active transport involves the movement of substances against their concentration gradient by requiring ATP. So energy is needed as an input in the form of ATP to get substances in and out of the cell. So active transport, it moves substances from the region of from the region of lower concentration to the region of higher concentration with the use of energy in the form of ATP. Then this active transport this process is essential for maintaining cellular concentration of ions and nutrients. So active transport allows cells to accumulate necessary substances despite external conditions. So active transport allows cells to accumulate necessary substances despite there is an external condition. So you see, this is a typical example. You see, in this case, the molecules are moving from the region of, like, look at this blue ball. The concentration of blue ball is lower from outside here, from outside. So it is moving from the region of lower concentration to the region of higher concentration using ATP. And of course, look at the red ball. The red ball here have a higher concentration outside of the cell, and then inside of the cell, their concentration is very low. So that is why they are getting out of the cell. So they are moving from inside of the cells to outside of the cells, because in the inside of the cells, the red ball have a lower concentration, and outside of the cell, the red ball is having a higher concentration. So the movement of these molecules, like for example, the red color is glucose. So you can see that the glucose molecules have a higher concentration outside and lower concentration inside. So it is moving from in to outside. Of course, energy is needed That is why it is called active transport, because it requires energy for the transport. And of course, look at the potassium and sodium at the same time. So you see, potassium have a lower concentration, and as they are getting in into the cell, and at the same time, the sodium ions is getting out of the cell. So this is a typical examples of the active transports. So there are basically two types of active transport. We have primary active transport and secondary active transport. So in the case of primary active transport, directly it uses ATP to transport molecules such as sodium, potassium form. So it uses ATP and moves a molecules across the cell membrane, example sodium potassium form. And then secondary active transport, it relies on the energy created by primary active transport to move other substances. And that energy that was created by the primary active transport is electrochemical gradient. So the electrochemical gradient, which is the energy generated, is actually the energy that is needed to transport substances across the cell in the case of secondary active transport. So of course, this is a typical example of simple diffusion. Molecules are moving from the region of higher concentration to the region of lower concentration. And then in the case of facilitated diffusion, is also moving from the region of lower concentration to the higher concentration. But of course, we need, in the case of facilitated diffusion, there is a need for protein carrier and protein channel until equilibrium is attained. And also in the case of active transport, energy is needed, and it moves molecule from the region of lower from the region of lower to the region of higher concentration, and energy is needed. So these are the two types of active transports. Then the next types of transport is bulk transport. So a bulk transport is endocytosis and exocytosis. So endocytosis is the process by which cells engulfed external materials forming vesicles to bring them inside. So it forms a vesicles where engulfed the membrane, or the part of the cells engulfed external material, which lead to the formation of vesicles and then bring them inside. And in the case of exocytosis, is the reverse process where the vesicles containing material fused with the membrane to release their content outside the cell. So both processes are essential for bulk transport of large molecules. So large molecules like hormones and nutrients are transported across the cells using this bulk transport system, which is endocytosis and exocytosis. So let's look at this. So this is outside the cell and this is inside the cell. So in the case of the bulk transport, see, these are the molecules here outside. These are the molecules here outside. So you see, this is the membrane. So it will start holding to engulf this molecule. So once it engulfs it, then it will form a vesicle like this. So after forming the vesicles, then it will now bring the vesicles inside of the cell. Then it will now cut off and then release the contents. So that is in the case of endocytosis and exocytosis. Vesicles are formed which will bring the molecules inside or bring the molecules outside, depending on the types of the molecules, the size of the molecules. Then, of course, these two transport are very important in bulk transport of materials like hormones and nutrients. So, the next thing after this endocytosis and exocytosis is conclusion. Please, ladies and gentlemen, I wanted you to join and try to subscribe to this YouTube channel. So conclusion and importance of membrane transport. So the transport of materials across the cell membrane is fundamental for cellular communication and metabolism. So transport of substances to get into the cell is very, very important in cellular communication. Like, for example, if a virus gets into a particular cell, if it transported into a particular cell, then of course it's through that transport that will actually, that cells will try to communicate other cells by releasing a molecules that will actually be get uh, communicated by the other cells. So it's through molecules that other cells will be communicated by the infected cells with virus or bacteria. And of course, in the case of metabolism, so glucose will be digested, sorry, will be transported from blood to the cells. And of course, from outside of the cell, it will get into that cells using a transport mechanisms before metabolism take place. And of course, disruptions in these transport mechanisms can lead to various disease and cellular dysfunctions. So if there is any disruption or any problem with the transport of molecules across the cell membrane, there will be a disease and cellular dysfunction. And remember, whenever there is a cellular dysfunction, it means that there will be a disease. So understanding these processes is crucial for developing medical treatment and advancing biotechnology. So that is why understanding these transport mechanisms is very, very important in the field of medicine and biotechnology. So these are some of the references. Please, thank you very much. And if you didn't join and subscribe to this YouTube channel, please try to subscribe. Thank you. Thank you. Thank you. Thank you very much for your time.